So good morning, and thanks to Sudipto for giving this topic to me. I think it's an honor to speak here in front of Dr. JP, who's been our teacher during UG days and learned a lot of epidemiology study design from sir, and so it's nice to speak in his presence. So I'll be talking about how do you interpret graphs in microbiome research, So obviously, at the last 2 decades, there's been a flurry of work on gut microbiota, and it has been almost any disease you think of, GI, non-GI, there have been some link with gut microbiota. So the amount of research which is happening on gut microbiota has really increased tremendously. And we know that the GI tract contains a large number of organisms, which includes bacteria, fungus, virus. And most of the bacteria actually are anaerobes, which are difficult to culture. So previously, when we were relying on the culture-based techniques, we were only sort of looking at maybe 20%, 30% of them. Most of them were not recognized. So nowadays, the sequencing techniques are very popular, because with sequencing, you are able to sort of identify most of the organisms in the GI tract. So sequencing, again, are different types. Sometimes you do what is called as a marker gene, means you just sequence a very small fragment of the DNA, and that gives you an idea about what the bacteria is. So it's very cost effective, and so in the limited cost, you can do a lot of sequencing. Other one is you do the entire bacterial sequencing. So when you sequence the entire genome, you know about the function also, which is not possible with a smaller sequencing. So when you are looking at a microbiome paper, you should look at what is the methodology employed to look at the microbiome. Was it marker gene sequencing or shotgun sequencing, whole genome sequencing? Sometimes they have a qPCR also. So I think this slide here. So essentially what happens, so we'll basically talk about the molecular techniques or the sequencing methods. So you have a sample, stool sample or tissue sample, you extract the DNA, and then you do run it in the machine. So there are different platforms. What we use, Illumina, you have the Ion Torrent, you have the Nanopore, so many systems are there. And once you do the sequencing, this is how the reads will look. This is the read of just one of those extracted DNA fragments. So like this, you'll have hundreds and thousands of reads at the end of your sample run in the platform. So as you can see, this is one of our own study examples. So in this particular thing, we had more than 13 million reads after the end of the run actually, and about 50,000 per patient. So when you are having 50,000 such sequences per patient, millions of reads at the end of it, how do you analyze the data? How do you present the data? It becomes a bit challenging. And also when you see papers, most of journals, there's a limitation how many tables you can have, how many figures you can have. Usually about 6 to 8 is allowed. But the amount of data is so much that you have to be really careful into what you're putting in the main text. And a lot of these microbiome papers will also have the supplementary file where the other information will be put actually. And graphs are much more informative, gives you more information than what a table can give you. And one other way which people get around of this limitation of figures is that they use composite images. So one figure will have multiple images, so you can have more information. So often when you come across a paper on microbiome, you will see sort of images like this. So again, how do you make sense? What is this meaning of these different names, the Simpson, Chow, the phylogenetic, there's Unifrac here, there's LDA here. So at the end of my presentation, hopefully we can have some clarity about what these things represent. So as I told you, when you have this millions of sequences which you get at the end of run, you need to do some sort of first initial analysis and then the downstream analysis, and there are different pipelines available. The most commonly which we use is what is called the CHIME2 pipeline. So this does this, what is called as the demultiplexing. So you want to know which sequence came from which patient, you separate them out. Then you do quality control, you take out the poor quality reads. Then you put the similar sequences together. So this similar sequences together, when you put them together, it's called OTU picking. And then you want to know what that sequence is, which organism that sequence belongs to. That is called as assigning the taxonomy. You also want to know how these organisms are linked to each other. Are they close to each other? Are they far apart from each other? So you have this phylogenetic tree. And once you've done all the analysis, then we are ready for the downstream analysis like alpha diversity, beta diversity, what is the function. Some of them can be done in CHIME, but most often they use this R package, which has a lot of softwares linked to it. And many of the publication figures in journals you see would have been actually made in the R studio with the help of these packages like ggplot, etc. So what are the main things we are wanting to know when you do a microbiome study? We want to know who is there, like what are the organisms there. We also want to know what they are, how many of them are there, like how many types are there, what is the diversity, what the organisms are doing, that is what is the function. And finally we want to compare case versus control, one disease versus another disease. and then you want to know if microbiome can be a predictor, can be a prognostic marker. So for the first one, that what organisms are there, what taxonomy is there, often you will see these plots. These are called taxonomy bar plots. Now these plots can be for individual patient or it can be for the entire group. Like this is from our own example. So this plot is basically for each individual subject, and this is at the, so you have to look at what level the plot is. Is it phylum, is it family, is it genus or species? So this is at the level of phylum, and you can say like for this patient, the commonest taxa are firmicutes, bacteroids and proteobacteria. They make up more than 90%. So you can have individual patient taxonomy data, or there can be data for the entire group. Like for example, in this graph, each of this represents the entire group, the summary, the average of the entire group. Then this is at the phylum level, this is the genus level. So when you come to genus, the number of categories will actually increase. And then you can, so the two informations you get from this plot is, one is what is there, and second is how abundant they are actually. So these are two things you get at the individual and the group level from the taxonomy bar plots. Other way of representing it is what we call as a heat tree, taxonomy tree. So instead of bars, you have a tree-like thing. The central node is the bacteria. The next node is the phylum, then is the class, order. And the size of node tells you how abundant that is. For example, in this particular example of ours, firmicutes were the most common, followed by proteobacteria and bacteroids. So that's how you represent the taxonomy, or what organisms are there. The second thing we want to know, how many different types are there. This is one term which will come always in any microbiome paper as to what is the alpha diversity. So just to make it very simple, so we have examples of 4 samples here actually. Now in first sample there are only 2 types of object. In the second there are 4 types. So the second sample is more diverse compared to the first one. So this is basically what we in the this bioinformatics we call it more richness. So this sample is more rich compared to the first sample. If you look at the third sample, it has got 5 types of objects. So this is more rich than these 2. So richness means how many different types of objects you are having. The second thing is evenness. So if you look at this sample and this sample, they both have 5 type of objects, but in this one, the distribution is more even. Here the first 2 of them are actually taking up most of it, and others are pretty less in number. So if you have more even sort of distribution, the diversity is supposed to be better. So that concept is called evenness. So we have the richness, that is how many different types you have the evenness. And the third thing is the phylogenetic or the phylogeny, because you know the bacteria in evolutionary tree, some bacteria are close to each other, some bacteria are far away from each other. So like for in this example, bacteria 1 and 2 are closer and 1 and 5 are far away. So if a sample has 1 and 2, other sample is 1 and 5, the 1 and 5 will probably be more diverse in terms of phylogeny. So for alpha diversity, these are 3 metrics we use. What is the richness? What is the evenness? What is phylogeny? And based on this, there are too many different types of things which you will come across. All these names are difficult to remember. But you should look at what are they looking at. Are they looking at the richness, evenness, phylogeny? And some of them also use a combination of features like Shannon, etc., where the richness and evenness is actually combined. So if you look at this plot, this is a box plot. The median, this part is the median, this is the first quartile, Q1, the Q3, the upper and the lower end. Sometimes there may be outliers as well. So this tells you what is the median alpha diversity of one group, and the metric, like this is observed, this is the Chow, this is the Shannon, etc. So whatever metric they are using, what is the median diversity? So this is the usual way of depicting this diversity using box plots. The thing with box plots somehow is, but you know the median and quartile, but you don't know how the data is distributed. So a slightly better way of doing this thing is called as a violin plot. So in violin plot, you have the box, but you also depict the individual patient's data in this. It gives you some idea about how it is it uniformly distributed, are there multiple peaks of distribution. So violin plots also are commonly used to depict alpha diversity. So alpha diversity mainly by these box plots. So we discussed what is the taxonomy, how many different types are there, which is alpha diversity. Next, we want to know what these organisms are doing, or what is the functional profile. Finally, what matters is what function they are doing. That will define the pathogenesis, or they are commensal organism, or they are beneficial, they are harmful. So the issue with function is, if you want to know the function, you want to know a larger gene sequence, you want to know about the RNA expression profile, you want to know the metabolites. So metabolomic, transcriptomic, or all this gene thing is actually more expensive. Most of the studies are looking only at 16S rRNA, which is looking only at a small fragment of the DNA. But even on 16S RNA, you have softwares like PyCrust, which can predict what the function of the bacterial thing is up to 80% of accuracy. So when you're reading a paper on microbiome, you see how the function was. Was it by the full genome sequencing, metabolomic, or was it just a predicted function based on the PyCrust software? So for example, again for one of our papers, as you can see, this just tells you about the different pathways. So as you can see that the carbohydrate biosynthesis or the polypropylene biosynthesis, etc., is much more abundant than the other. So again, just the bacteria have so many different types of metabolic function. It just tells you which metabolic function is more abundant, which is less abundant in terms of this is a predicted function. So now we found out what the organism is, what is the diversity, what they are doing. Now we want to compare. We want to compare the cases with the controls and between the two groups. So comparison, there are now different metrics. The first is the beta diversity, again a very commonly used term. It's a little bit difficult to understand initially, but as you read more and more, you'll be able to get a clarity on this. So beta diversity is always comparison of diversity between two samples actually. So for example, if you have two samples, say site A and site B, the diversity within A is alpha diversity, but when you compare A with B, that is beta diversity. So if you look at this A and B, the black thing is there in A but not in B, the red is here in B but not in A. So if you say beta diversity, to put it very simply, the beta diversity will be 2 in this. Obviously there are very complex ways of calculating this, and imagine you are doing this comparison of one sample with each other one. So even if you have 50, the number of comparisons will be in hundreds and thousands actually. So therefore this again, there are different metrics. There is a breaker test, there is the unifrac. The unifrac takes into account the phylogeny, whereas the breaker test doesn't take into account the phylogenetic relationship. So when you do the distance calculation between objects, you get this sort of matrix. Now this matrix is not very easy to understand straight away, so people always try to make what we call as a plots. So these plots, there are different types. You have the principal component analysis, coordinate analysis, etc. Again, just to make it very simple, this is again one of our own work which we have done. So in this principal coordinate analysis plot, you'll see a lot of these dots. So each dot represents one particular sample. And then if you look at, so supposing there are these two dots here, so what it tells you that these two samples are very close to each other in terms of their diversity. But this dot is quite far away from this one. So it tells you how close the samples are to each other in terms of their diversity. Apart from that, you also have these circles which are drawn. Now if these circles or these curves which are drawn around the dots, if they are sort of overlapping, as you can see in this, then there is no visible difference in diversity. But if you look at this breaker test plot, there is a differential clustering of one group compared to the another group. So here the beta diversity shows there is a difference in diversity. We also want to know why is the difference. Is it only because of presence of disease or absence of disease? So there again you can do further analysis, and because microbiome depends on so many other factors, your diet, gender, region, etc. So for example, in this situation we had done a matching for age and gender, and what we found is the difference between case and control was because of two reasons. Diet was the most important factor, and second was the disease actually. So again, so disease is not the only one contributing to difference between the two groups actually. You can also compare the alpha diversity between the two groups by using the statistical tests, and we can compare the functions also. So whatever function is there, for example, in this one you can see that these three functions are more prevalent in the healthy patients and less in the control population. So essentially these are what we normally do. We look at what the taxonomy is, how many different types are there, what they are doing, how do we compare between the groups, and again just a couple of more things. Basically we also want to know, is this bacterial population able to predict between disease and control or prognosis or etc. So for here again, because there are so many bacterial taxonomy in your sample, you need something more than a normal statistical comparison. So often they use what is called as a machine learning algorithms, and one of the very common one is the random forest technique. So what random forest tells you is that it identifies which organism is linked with what particular situations. For example, you can say that what is linked with disease, what with health, what is active disease. And you can also represent data in the form of heat maps. Now heat maps are very commonly used in microbiome papers. They are usually color coded instead of numbered, so it's much easier to appreciate what is happening. So for example, you can say that this organism, bifidobacteria, is much more linked to a healthy state, whereas this organism is more linked with a disease state actually. Other way also we have this, let's say, or a linear discriminant analysis. This again telling you which organisms are linked with disease, which is linked with a healthy state. So again, all these ones are with one particular condition. So finally, when you have hundreds of organisms, you just want to know, so this is my final slide. So once you find the organism which is linked with disease and not with disease, you can make a ROC curve. This is again our work on TB versus Crohn's, whether microbiome can differentiate tuberculosis from Crohn's disease, work done by Ajit Kuryakos, one of my thesis students. So what we found is that the microbiome groups can help you to differentiate tuberculosis from Crohn's, and you get area under the curve and all that thing. But again, you look at what the 95% CI is, and obviously you know that the CI is wide, so it may not be very applicable. So again, so like this, there are so much of plots in microbiome study, and this is not everything, but I have told you about the common ones. So in this figure, just coming back to it, so these two are the taxonomy bar plots. The one is at the phylum level, this is at the family level. All these are the box plots at the alpha diversity. They have used about 8 alpha diversity in this particular thing. These figures you are seeing, this is actually beta diversity, and you can see there's a lot of differential clustering, which means diversity is different, and they have used the unifrac metrics here. And this is the LDA curve showing these predict the MH state, these predict the non-MH state. Thank you for your attention.